It now gives me a great pleasure to introduce Michele De Luca. Michele De Luca is professor of biochemistry and director of the stem cell and regenerative medicine at the University of Modena and Reggio Emilia. He's a doctor, he's an endocrinologist, but he devoted his life to cell biology, particularly epithelial cell biology, and lately to stem cells. With all the news, all the discussion that you see in newspapers about stem cell misuse, he has been taking a very strong ethical position. And if you read the Domenicale of the Sole 24 Ore, you can see a lot of his articles, emphasizing the need of science in this field. And also has been the pioneer in application, medical application of stem cells. So I asked him to illuminate us, to give us his perspective on what the state of the art of the potential of stem cells in. Thank you for accepting our invitation.
Thank you, Leo. And now I finally understood why he invited me. Because at least you know why you've been invited, but I didn't know. Because what I'm going to talk to you about today is not related to anything you do, and it's going to be so difficult to bridge with anything else. I've heard so. It's going to be a challenge. But anyway, I mean, the field of the stem cells today is quite interesting. Today I'm going to speak about the epithelial stem cells, and you understand why. The main reason is because we do it, but
That's what I do for business.
The second reason is that everybody speaks about stem cells, but what is into the clinics today, there are only two stem cells, which is the hematopoietic and the epithelium. So, regenerative medicine today refers to advanced therapies that have the aim of restore or regenerate a tissue that is lost. And because of most of the tissues we have are self-renewing, we change our blood every 3 months, our skin every month or so, or the cornea every 6 or 7 months, we do need stem cells in order to renew those tissues. So if we want to recreate a tissue which is destroyed, we have to have the stem cells. But saying stem cells today, it doesn't mean much because we have a huge number of stem cells. And the mother of all stem cells is the embryonic stem cells in the inner cell mass, in the embryoblast. And the embryonic stem cell is the only pluripotent, naturally occurring, pluripotent stem cells we have in nature, no matter what David Evannoni and Stamina says. And during the development, the embryonic stem cell give rise to ectoderm, mesoderm, and endoderm, and so all the tissues, which contain specific stem cells, which are called somatic stem cells. Those are bona fide. By all means, they are stem cells because without them we die, okay? And those are responsible for the generation and the regeneration of the tissues where they are. But they are not pluripotent, they are multipotent. There are 3 examples here, but there are many. For instance, the epidermal stem cells give rise to the hair follicle, the interfollicular epidermis, the sebaceous gland. Historically, the hematopoietic stem cells, one of the best characterized stem cells, give rise to all the elements of the blood, or the intestine give rise to all the cell types we have in the intestine. It is also true that we can reprogram a stem cell. It doesn't mean a stem cell is plastic. A somatic stem cell is not plastic. I cannot make blood out of my epidermal stem cell, and I cannot make skin out of the hematopoietic stem cell. So they're not plastic. They can be reprogrammed by genetic engineers. So you have to add master genes in a cell in order to make reprogramming. And we know this since the 60s, when the frog has been cloned by Gurdon, and by the 80s, where the Dolly the sheep was made by Jan Wilmot. What Shinya Yamanaka did a few years ago was to find out 3 or 4 essential reprogramming factors that can make what is called the iPS. So the iPS is a pluripotent cells that then can be redirected in vitro to make all this tissue. And the important thing of the iPS is not because we don't touch the embryo. The important thing is because those cells are autologous. And therefore, if we want to use one of these cell types that are arising from an iPS, we don't have to worry about immunogenic reaction. There are only 2 cell types that are into the clinics. Why? Because the embryonic stem cell is in clinical trial. There are only, to my knowledge, 2 clinical trials in the world running for embryonic stem cell, for spinal cord injury and for macular degeneration. There is 1 clinical trial that's been approved in Japan for the iPS, again for retinal disease. There are approximately 350 clinical trials in the world, but I will talk about this at the end of my talk if I have time, concerning the mesenchymal stem cells. Again, clinical trial phase 1 or phase 2, and we have no idea about the data. What is into the clinics, so what has been shown to help people in curing anything, are only the hematopoietic stem cell and the epithelial stem cell. The hematopoietic stem cell is the historical application of a stem cell. It's been the first cell therapy, the first stem cell has been applied into the clinics. And then it's been done by Edward Donald Thomas in a New England Journal of Medicine paper in 1957. He won the Nobel Prize for this, and is a famous bone marrow transplantation that we use to infuse not the mesenchymal stem cell, but the hematopoietic stem cells in order to treat leukemia, plus a number of other diseases of the blood. Not many people know that the first cultivated, amplified in vitro, cultured cell type that has been applied into the clinics is the epidermal stem cell, done by this guy, Howard Green, that was my mentor the time I was a couple of centuries ago in Harvard, when he was developing this. And it still is, those 2, the only established worldwide use therapy for a stem cell. So there is a long way to go. What I'm going to talk to you today is a few things that we have developed lately in our lab with cell therapy, and also I'm going to speak about the genetic modification of those stem cells if we want to use stem cells in gene therapy. Now, if we take a skin biopsy or a cornea biopsy or any biopsy containing a squamous epithelia, those cells, they make colonies, they grow, and you can detach the epithelium that you can culture in the bottom of the flask by an enzyme called dispase. If you study stem cells today, 2014, you have to go at the clonal level. Nobody's going to believe you anymore if you pretend to describe, to characterize a stem cell in a mass population. You have to clone the cells. And if you clone the cells, so if you grow the cells at a single cell level, you will see that you have a number of clonogenic cells in the skin, like in the cornea. They are called the activated stem cell, the quiescent stem cell, and the transient amplifying progenitors. They all give rise to colonies. And you will see how this is going to be important from a clinical point of view. Because only about 5% of the clonogenic cells are stem cells, and they are called holoclones. And the holoclones are the stem cells of all squamous epithelia. So the vast majority of the clonogenic cells we have in an epithelium are transient amplifying progenitors, like, for instance, the common myeloid progenitor or the common lymphoid progenitor we have in the blood that give rise to the blood elements. And the holoclone has all the characteristics of the stem cells. I'm not going to go into this, it's going to take too much time, but all the characteristics of the stem cells, more importantly, is important for permanent tissue regeneration in humans, as has been shown for the skin burns. Today I'm going to summarize in a few slides a long story that we started many years ago when I was still in Genova at the time, together with Ranieri Canceda, an ophthalmologist called Carlo Traverso, which is the regeneration of the cornea. And we published the first proof of principle many years ago in the Lancet. Then a collaboration started with a wonderful corneal pathologist, which is called Panorama, at the San Raffaele Hospital, also in collaboration with other ophthalmologists in Rome, Alessandro Rambiase, Stefano Bonini, but most of the work has been done by Panorama, to try to bring into an established clinical protocol, reproducible and formally proven to be efficacious and safe for the corneal regeneration. And this was the paper we published in 2010. Meanwhile, we were trying to apply this with multi-center experience with many ophthalmology departments. We just published a long-term study with a number of ophthalmology departments. All of this work has been done by Graziella Pellegrini. We have been working together since many years. She started to develop this years ago when we started working together for the cornea. That was a really important project. So she got a lot of job offers around, and I did the only thing I could do to keep her in the lab. So I married her, and and we did immediately one kid, so she couldn't move anymore. Now, if you look at the ocular surface is, the cornea is transparent, is not vascularized, the conjunctiva is heavily vascularized. They have markers like keratin 3 and keratin 19. This area here, which is called the limbus, it's important is to understand all the clinical application of this technology. This rim of cells that is called the limbus is about 1 millimeter wide. That is at the border between the cornea and the conjunctiva, contains the stem cells of the corneal epithelium. So those stem cells are in a niche, completely segregated from the transient amplifying progenitors, which are these pink cells that during constant renewal of the cornea, they generate out from the stem cells, they make the basal layer of the corneal epithelium. So in the limbus, we have the stem cells. In the basal layer of the cornea, we have only the progenitors. So they have a limited proliferative capability. If we have an ocular burn, chemical burn, destroying the cornea, but not the limbus, not completely the limbus, what happens is that the stem cells in the limbus get activated immediately. It is a matter of minutes of hours. They proliferate, they go into the lesion, they make up a corneal epithelium, and they restore the epithelium that's been damaged. However, most of the time, you have stromal scarring. The corneal stroma is transparent because the collagen is made in a specific way. So if you have a stromal scarring, you have situations like this, when you have a central corneal opacification with ulceration and stuff, but you have still the limbus. This is the vast majority of the cases. And if you have a situation like this, when you have total opacification of the central part of the cornea, but the limbus is still okay, what you usually do is a standard keratoplasty, is a corneal transplantation. You remove the central part of the cornea, you take a donor cornea, you transplant the donor cornea in the lesion, and this technology works fine. Why? Because the aim of the keratoplasty is only to restore the stroma. But the epithelium of the patient, will cover the donor stroma. That's why the keratoplasty works. You have situations like this, though, when you destroy everything, cornea and limbus. Therefore, what is happening here is that the conjunctiva is invading the central cornea, is bringing vessels, and is causing what is called corneal opacification or corneal conjunctivalization. There is no limbal corneal epithelium anymore. The eye is invaded by the conjunctival epithelium. It gets vessels. You get inflammation, neovascularization, opacification, severe clinical symptoms, lots of vision. This is a rare condition. It's a rare disease. It's approximately 1 out of 300,000 patients, so it's a minority of the chemical burden of the eye. But they have no real alternative therapy, because if you take an eye like this, in which you have total complete opacification and neovascularization, and you make a keratoplasty, it will never work because you don't have resident residual limbal stem cell to stimulate. And therefore, the keratoplasty fails, the inflammation goes worse, and in many cases, you have to take out the eye and put an eyeglass. Now, with the experience we have in the skin, what we thought at the beginning was incomplete unilateral lesions like this, in which you have a total limbal stem cell deficiency, or in severe bilateral lesion that cannot be treated with a keratoplasty, but they still have some limbus left. We could grow autologous limbal cells, keep the holoclones, and I will go back to this, and try to transplant this back into the patient. The surgery, it is actually quite simple, because this is the 1 millimeter, it's just a fragment, it's 1-2 millimeter limbal biopsy that is taken from the LTI on day 1, and this is the fragment we have in the lab where we grow the holoclones. About 3 weeks later, what Paolo Ramain is doing right now is to remove, under anesthesia I hope, is to remove entirely the pannus, entirely the conjunctival epithelium, and to expose the stroma. And this is the graft, on fibrin in this case, containing the cells and the stem cells, that is applied on the surface. Okay? The edge of the graft is flipped under the conjunctival flap, is about 1 stitch, it's about 1 hour surgery. And the ophthalmologist gets the entire credit for it, the entire technology, but we are used to it. But let's see the results. Those are the eyes we're talking about. You see, complete opacification, neovascularization, 360 degree. A sign of a keratoplasty that failed, no alternative therapy, severe symptom, loss of vision. Those are the eyes after the regeneration done with the cultures of limbal stem cells, 2-4 years follow-up, full recovery of visual acuity. Sometimes we have to combine the cells with the keratoplasty because of the stroma, but it's going to be too long, to explain this. Look just at the eye. Those are, again, eyes that are completely damaged, opaque and vascularized, a sign of keratoplasty that failed. Look at the eyes, 5, 6, 7, 10 years follow-up, very stable, full recovery of visual acuity. In this study that I was referring to, 154 patients in 10 ophthalmology departments, 14.5 years follow-up, median of 8 years. So remember, we change our cornea every 7 months. So either we transplant the stem cells, or we could not have corneal regeneration for a long time. All patients had severe symptoms. Most of them, they were male. Vast majority chemical burns. Vast majority were trying to do all sorts of surgery without benefit for the standard therapy. And 75% of those they have a very negligible visual acuity, so on movements or light perception. We got about two-thirds, 66% of the patients that have a full success. That means the eye I showed you before. We still have one-third of patients, approximately, altogether, that is not working fine. The technology, we have complete failure in 15% of the patients, about 20% partial success. The eye gets better, but it's nothing compared to what I've shown before in the eyes. So we want to understand what's going on here. Why is working so nicely in two-thirds of the eye? Why we are losing eyes in such an amount? So we have to go back to the lab, probably. And in fact, if we do a couple of male estimates, a survival rate, you see that the failures are concentrated in the first 6 months. The moment you score a patient as a success, it is stable for his entire life. So something is happening here. And the first thing that came into our mind was an insufficient number of stem cells in the culture. Why? Because we are starting with a very small biopsy containing no more than 20 stem cells to start with, okay? But we have no marker at that time, and cloniformic efficiency, it is not a marker. There was no difference in the number of clonogenic cells in success and failure. The colony size was not a marker. Stem cell and progenitors, they have the same size. The growth rate was not a marker. So cultures that failed and were successful, they were growing identically. Regenerative medicine required basic science. So it took us years and a number of papers to try to characterize at the molecular level the stem cells. And we find out 3 transcription factors, delta N p63 alpha, CBP delta, and BMA1, that were identifying and distinguishing the stem cells from the transient amplifying progenitors. P63 regulates proliferative regenerative potential, CBP delta self-renewal, and mitotic quiescence. But those are transcription factors. So we cannot identify the cells based on a protein that is in the membrane. So we had to validate the level of expression of P63 by quantitative automated fluorescence immunohistochemistry we developed together with Zeiss. And finally, we are able to know how many stem cells we have in the graft before clinical application. So we went back to our patients and see that finally we found a correlation because the number of stem cells was significantly higher in the success rate than in the failure. More importantly, in the failure you have a median here, which is 3%. So we analyzed the data in a different way. We took all the cultures containing more than 3% of stem cells, that means 3,000, and less than 3%, and see what was the clinical outcome. And that was really straightforward. If you have about 3,000, there is a reason for this, if about 3,000 stem cells, at least 3,000 stem cells in the graft, you have a success, a very little failure. The moment you drop below the number of the threshold of 3,000 stem cells in the graft, then the success rate falls down dramatically and the failure goes up. And if we have time in discussion, we will address this, why this happens. Okay? This was done in one study done by Panorama, the one published in the New England, in one ophthalmology department. This has been reproduced in another paper where we have 10 ophthalmology departments participating. So the data is quite solid because it is really superimposable the results we have, both in terms of percentage of success and failure, and both in terms of the number of stem cells in the graft. That is the way I see where regenerative medicine with stem cells should be done. And if I have time, we can address the stamina issue at the end. In the last few minutes, I want to talk about the gene therapy. Because once you have the stem cell in your hand, you know how to apply in cell therapy, and therefore you know how to regenerate the tissue after transplantation. Then you can take care of the problem of gene therapy. And obviously, the only two cell types, stem cells, has been proven to be clinically effective in gene therapy has been, again, the hematopoietic stem cells. This is a pioneering work done by Alessandro Aiuti. Italy is quite strong in this field, in general, of regenerative medicine. This is a paper by Alessandro Aiuti and Claudio Bordignon. Is a long-term study of a gene therapy of an immunodeficiency due to adenosine deaminase. Is the ADAS kids, is the kids that live under the bubble. And all these kids has been saved by this gene therapy trial, and the long-term follow-up is almost 10 years. And this is the first clinical trial has been done outside the blood. It's been done by us in collaboration with Mavilio and the dermatology department of Modena on the gene therapy using epidermal stem cells. Which kind of pathology? Epidermolysis bullosa. Epidermolysis bullosa is the butterfly syndrome. These kids, they have gene mutation in each of the protein that links the epidermis to the dermis. They have the simplex form, junctional form at this level with laminin 5, integrin alpha 6 beta 4, which is involved, and the dystrophic type, which is involved collagen 7. Very severe disease, very poor quality of life, devastating blistering, short life expectancy. We got the authorization by the national authorities to do a clinical trial with ex vivo gene therapy on one patient only, and only to selected area of the legs. The patient is this guy, Claudio, which is a compound heterozygous carrying a frame shift mutation, so a PTC1 allele, and an amino acid substitution in another allele on the beta 3 chain of laminin 5. The skin of Claudio is like this in the entire body. What we did was we took a biopsy from the palm because we had an area particularly rich in Claudio in stem cells. We used the very same vector that Alessandro Aiuti was using for the other deficiency in the blood. So we transduced the holoclones just to show you by southern blot, northern blot, by immunoprecipitation that the genetic defect has been corrected in the stem cells. Go fast on this. And the area of the skin has been prepared, has been removed all the remnants of the diseased epidermis, and the graft composed of transduced epidermal stem cells were applied here and regenerated the normal skin. This was about, it's a little bit less than 1 year follow-up. These green dots is an in situ hybridization showing that the cells contains the transgene. So they contain the new gene that we have introduced with the virus into the genome. This is the immunofluorescence of laminin 5 of Claudio before the transplantation. You see, is negative. There is no expression of laminin 5, and that's the reason why he makes the blisters. This is the expression of laminin 5, how it is supposed to be in the normal control, and this is Claudio in the right, in the left leg at 1 year follow-up. So the gene therapy trial was working fantastically. So we were ready to do the remaining part of the body of Claudio and to try to include new patients in the trial. But the European community established this EU regulation, 1394 in 2007, that basically stopped completely the entire field of regenerative medicine. Because from that point. Was stated in the law that all cultured cells, no matter whether it's gene therapy or not, they are medicine. And therefore they fail under the regulation of drugs and should be done like drugs. Therefore, under GMP, GCP regulations. So after 23 years of clinical application and something like thousands of patients we have treated in the skin, in the cornea, I didn't mention the vitiligo, the urethra. We had to stop everything, and we have to do this, to build up the GMP facility. So this is the reason and why the Center for Regenerative Medicine of our university was born, thanks to Stefano Ferrari, who was the dean of the faculty at the time I was, and he died 2 months before the opening of the center, and was entirely financed, totally financed by the Banca Foundation, la Fondazione Cassa di Risparmio di Modena. But then we had to do GMP certification of the facility. And the university does not have the expertise, it doesn't know how to do it. So finally we found a partner, again in Emilia Romagna, which is Chiesi Farmaceutici, is a big company, and the university made a university spin-off, Orostem, with Chiesi, and Chiesi is taking care, or he took care, of the GMP certification of the entire Center for Regenerative Medicine. As a matter of fact, we also need more authorization for continuous hour therapy, but from 2006 to 2012, we couldn't do any patients anymore. In 2012, we started again with the cornea. And now we are planning to resume our gene therapy clinical trial, not only in Italy, not only in Modena, but also in Salzburg, at the University of Salzburg, in a month or so. In April, we should take the first biopsy for the new patients. But meanwhile, Claudio has 7 years follow-up. So in gene therapy, 7 years is quite important. So we were able to analyze Claudio after 7 years. Not a single blister, not a single blisters in the area. The entire epidermis is transgenic, because this is an in situ hybridization, and you see that the messenger RNA is there. So the skin that is in Claudio is the one we put 7 years before, transgenic. No difference whatsoever between Claudio and the normal control for all the adhesion protein. So the adhesion machinery of Claudio, all the 3 chain of laminin 5, the integral alpha 6 beta 4 has been totally restored. Look at this. In the palm and in the sole, there is a specific keratin, keratin 9. It is only in the palm and in the sole, epidermis. After 7 years, Claudio is still expressing keratin 9 in the leg. So the stem cell of the palm was not even able to switch the program to leg epidermis. It was still keeping the memory of a palm epidermis. Can you imagine how easy it would be to make spontaneously a motoneuron out of a mesenchymal stromal cells? That's what people pretend to do in the stamina story. The last thing is that the cells are genetically labeled. So we could actually count the cells we had in the skin of Claudio, and we have calculated that we have approximately 1 stem cell per millimeter square of the regenerated transduced epidermis. Now, with this, which is 25 minutes, I finished. If you want, we can speak about 2 or 3 minutes on the stamina story. Very briefly.
Yes.
Very briefly.
Yes, okay.
Yes.
There is
Very briefly. Now, the stamina case, it will take 2 hours. But I don't think that anybody knows what is really behind this. And I try to be very fast in 2 or 3 slides, because all the interaction with the government is something that will require hours. The stamina method is an infusion of allogeneic or autologous. It doesn't matter. Mesenchymal stem cells to treat any kind of pathology you can think of. Marino Andolina 3 months ago said, I will revolutionize all the medicine and 80% of all the human disease will be treated by intravenous infusion of mesenchymal stem cells. If you inject a mesenchymal stem cells in the blood, you get the instant blood immune reaction, instant blood immune reaction. The cells are killed, and the ones that survive, they get trapped into the pulmonary circle. Now, the stamina method, they thought that they were making neurons out of mesenchymal stem cells in about 2 hours. For a cell biologist, making a neurons out of a fibroblast with no gene reprogramming in 2 hours is ridiculous. Who is proposing this therapy? Davide Vannoni. I don't know if you know that Davide Vannoni has a degree in humanities. He was associate professor of psychology, and the fact he was because he was fired, University of Udine, and he wrote a book on persuasive communication. So Italy is putting a clinical trial in the hand of this guy that does not have any idea what a cell is. Behind this guy, there is a company, Medestea. Medestea has been prosecuted 25 times for pubblicità ingannevole. Come cazzo si dice pubblicità ingannevole?
False. False.
25 times. You know why? Because a culture of mesenchymal stem cell costs about 30,000 euro. They claim that they can treat all the 8,000 rare disease. It means 1.5 million of patients in Italy, with a potential market of 45 billions of euro. That in the mind of Medestea, Merizzi and Vannoni should be paid by who? By the Servizio Sanitario Nazionale, by the health system in Italy. Okay? Just to tell you that these are the diseases that officially they said can be cured by the infusion of fibroblasts, which are the mesenchymal stromal cells they infuse. I don't know if you realize what is written in there. It's unbelievable. Look at this. Autism. Autism.
Autism.
Is the pontocerebellar hypoplasia. Defect in the development of the body can be cured by an infusion of fibroblasts, of mesenchymal stromal cells. The vast majority of this disease are neurodegenerative. You name one, is there. The most crazy to me is SMA1. SMA1 is a genetic disease of the motor neurons. How the hell can a mesenchymal stromal cell that makes three things, cartilage, bone, and fat. This is what the mesenchymal stem cell does. Cartilage, bone, and fat. How can it cure a genetic skin disease? Now, this is not an Italian problem. This is a global problem. This company here, is called XL, was doing the same thing in Germany. But there was a German government, the German police. So this company was closed. Celtex was done in the US. The guy ran away in Mexico and the FBI was running like a dog after him. And now he wanted Mexico because he was able to cross the border. And there are many examples of this. There are companies like this that are stealing money to people only in places like Panama, Mexico, China, Thailand, all these places. So the real question is, how was it possible after that in 2012, AIFA and NAS and Carabinieri were closing Stamina in Torino, that everything started again in a public hospital like Brescia, at the government level in our Ministry of Health, where a minister called Balduzzi was almost approving a stamina law that was basically making legal this type of garbage, transforming, because it was a compassionate use, the culture of mesenchymal, only mesenchymal, from a drug into a transplant, avoiding therefore the possibility or the necessity of demonstrating phase 1, phase 2, phase 3 clinical trial. That is the real question. What are the connections that made it possible to have these things going so on in our country, in a public hospital, in a region like Lombardia, and at the government level? There are investigations running on this, but this is a very important point to avoid future things like this. Sorry for the time.
Okay.
Thank you.
Thank you.